Tonight's lecture is over DNA transcription and translation. Let's look more closely at transcription, that is the transfer of genetic information from DNA into RNA. An RNA molecule is transcribed from a DNA template by a process that resembles the synthesis of a DNA strand during DNA replication. The figure on the right side of the slide shows a close-up view of transcription. As RNA nucleotides base pair one by one with DNA bases on one DNA strand, called the template strand, the enzyme RNA polymerase links the RNA nucleotides into an RNA chain. As with replication, the two DNA strands must first separate at the place where the process will start. In transcription, however, only one of the DNA strands serves as a template for the newly forming molecule. The nucleotides that make up the new RNA molecule take their place one at a time along the DNA template strand by forming hydrogen bonds with the nucleotide bases there. Notice that the RNA nucleotides follow the same base pairing rules that govern DNA replication, except that U rather than T pairs with the A. The RNA nucleotides are linked by the transcription enzyme RNA polymerase, or we sometimes call that RNAP. Again, the figure on the right-hand side of your screen is an overview of the transcription of an entire gene. Special sequences of DNA nucleotides tell the RNA polymerase where to start and where to stop the transcribing process. I would also like to point out that there are 3 steps to transcription. That is the initiation of transcription, RNA elongation, and the termination of transcription, and we'll discuss those steps in more detail on subsequent slides. Moving on, in the cells of prokaryotes, which lack nuclei, the RNA transcribed from a gene immediately functions as messenger RNA, or mRNA, the messenger molecule that is translated. But this is not the case in eukaryotic cells. The eukaryotic cell not only localizes transcription in the nucleus, but also modifies or processes the RNA transcripts before they move into the cytoplasm for translation by the ribosomes. One kind of processing is the addition of extra nucleotides to the ends of the RNA transcripts. These additions, called the cap and tail, protect the RNA from attack by cellular enzymes and help ribosomes recognize the RNA as mRNA. Another type of RNA processing is made necessary in eukaryotes by non-coding stretches of nucleotides that interrupt the nucleotides that actually code for amino acids. It is as if unintelligible sequences of letters were randomly interspersed in an otherwise intelligible document. Most genes of plants and animals, it turns out, include such internal non-coding regions which are called introns. The coding regions, the parts of a gene that are expressed, are called exons. Both exons and introns are transcribed from DNA into RNA. However, before the RNA leaves the nucleus, the introns are removed and the exons are joined to produce an mRNA molecule with a continuous coding sequence. This process is called RNA splicing. RNA splicing is believed to play a significant role in humans in allowing approximately 25,000 genes to produce many thousands more polypeptides. This is accomplished by varying the exons that are included in the final mRNA. With capping, tailing, and splicing completed, the quote-unquote final draft of eukaryotic mRNA is ready for translation. Moving on, as we have already discussed, translation is a conversion between different languages, from the nucleic acid language to the protein language, and it involves more elaborate machinery than transcription. The first important ingredient required for translation is the mRNA produced by transcription. Once it is present, the machinery used to translate mRNA requires enzymes and sources of chemical energy such as ATP. In addition, Translation requires two heavy-duty components, ribosomes and a kind of RNA called transfer RNA. Translation of any language into another language requires an interpreter, someone or something that can recognize the words of one language and convert them to the other. Translation of the genetic message carried in mRNA into the amino acid language of proteins also requires an interpreter. To convert the three-letter words or codons of nucleic acids to the amino acid words of proteins, a cell uses a molecular interpreter, a type of RNA called transfer RNA, abbreviated tRNA. A cell that is producing proteins has in its cytoplasm a supply of amino acids, but amino acids themselves cannot recognize the codons arranged in sequence along with messenger RNA. It is up to the cell's molecular interpreters, tRNA molecules, to match amino acids to the appropriate codons to form the new polypeptide. To perform this task, tRNA molecules must carry out two distinct functions. One is to pick up the appropriate amino acids, and two is to recognize the appropriate codons in the mRNA. The unique structure of tRNA molecules enables them to perform both tasks. A tRNA molecule is made of a single strand of RNA, one polynucleotide chain, and it consists of about 80 nucleotides. The chain twists and folds upon itself, forming several double-stranded regions in which short stretches of RNA base pair with other stretches. At one end of the folded molecule is a special triplet of bases called an anticodon. The anticodon triplet is complementary to the codon triplet on mRNA. During translation, the anticodon on the tRNA recognizes a particular codon on the mRNA by using base pairing rules. At the other end of the tRNA molecule is a site where one specific amino acid attaches. Although all tRNA molecules are similar, there are slightly different versions of tRNA for each amino acid. Ribosomes are the organelles that coordinate the functioning of the mRNA and tRNA and actually make polypeptides. A ribosome consists of two subunits. Each subunit is made up of proteins and a considerable amount of yet another kind of RNA called ribosomal RNA or rRNA. A fully assembled ribosome has a binding site for mRNA on its small subunit and binding sites for tRNA on its large subunit. Two tRNA molecules get together with an mRNA molecule on a ribosome. One of the tRNA building sites, the P site, holds the tRNA carrying the growing polypeptide chain, while another, the A site, holds a tRNA carrying the next amino acid to be added to the chain. The anticodon on each tRNA base pairs with a codon on the mRNA. The subunits of the ribosome act like a vise, holding the tRNA and mRNA molecules close together. The ribosome can then connect the amino acid from the tRNA in the A site to the growing polypeptide. And on this side, we'll dig a little deeper into the translation machinery. Ribosomes are complex macromolecules composed of structural and catalytic rRNAs, and they have many distinct polypeptides. tRNAs are structural RNA molecules that were transcribed from genes by RNA polymerase III. They actually, quote-unquote, translate the language of RNA into the language of proteins. Furthermore, each tRNA molecule is linked to its correct amino acid by a group of enzymes called aminoacyl tRNA synthases. At least one type of aminoacyl tRNA synthase exists for each of the 20 amino acids. Moving on, during the 1960s, scientists cracked the genetic code. This is a set of rules relating nucleotide sequences to amino acid sequences. As a preface to the next slide, 61 of the 64 triplets code for amino acids. The triplet AUG has a dual function. It not only codes for the amino acid methionine, but can also provide a signal for the start of a polypeptide chain. 3 other codons do not designate amino acids. They are the stop codons that instruct the ribosomes to end the polypeptide. Almost all the genetic code is shared by all organisms, from the simplest bacteria to the most complex plants and animals. The universality of the genetic vocabulary suggests that it arose very early in evolution and was passed on over the eons to all of the organisms living on Earth today. Such universality is extremely important to modern DNA technologies. Because diverse organisms share a common genetic code, it is possible to program one species to produce a protein from another species by transplanting DNA. This allows scientists to mix and match genes from various species, a procedure with many useful applications. Besides having practical purposes, a shared genetic vocabulary also reminds us of the evolutionary kinship that connects all life on Earth. And this next slide shows the dictionary of the genetic code listed by RNA codons. Notice that there is redundancy in the code, but no ambiguity. For example, although codons UUU and UUC both specify phenylalanine, that's a redundancy, neither of them ever represents any other amino acid, so therefore no ambiguity. The codons in the figure are the triplets found in RNA. They have a straightforward complementary relationship to the codons in DNA. The nucleotides making up the codons occur in a linear order along the DNA and RNA with no gaps separating the codons. Practice using this dictionary by finding the codon UGG. This is the only codon for the amino acid tryptophan. Notice that the codon AUG, highlighted in red, not only stands for the amino acid methionine, but also functions as a signal to start translating the RNA at this place. 3 of the 64 codons, which are in boldface black, function as quote-unquote stop signals that mark the end of a genetic message, but do not encode any amino acids. So as I said on a previous slide, we were going to dive deeper into the translation process. So translation is divided into the same 3 phases as transcription, that is initiation, elongation, and termination. The first phase brings together mRNA, the first amino acid with its attached tRNA, and the 2 subunits of a ribosome. The mRNA molecule, even after splicing, is longer than the genetic message it carries. Nucleotide sequences at either end of the molecule are not part of the message, but along with the cap and tail in eukaryotes, they help the mRNA bind to the ribosome. The initiation process determines exactly where translation will begin so that the mRNA codons will be translated into the correct sequence of amino acids. Initiation occurs in 2 steps. Firstly, an mRNA molecule binds to a small ribosomal subunit. A special initiator, tRNA, then binds to the start codon, where translation is to begin on the mRNA. The initiator, tRNA, carries the amino acid methionine. Its anticodon, UAC, binds to the start codon, AUG. Secondly, a large ribosomal subunit binds to the small one, creating a functional ribosome. The initiator, tRNA, fits into the P site on the ribosome. Once initiation is complete, amino acids are added one by one to the first amino acid. Each addition occurs in a 3-step elongation process. In the elongation process, the first step is codon recognition. The anticodon of an incoming tRNA molecule carrying its amino acid pairs with the mRNA codon at the A site of the ribosome. Secondly, peptide bond formation occurs. The polypeptide leaves the tRNA in the P site and attaches to the amino acid on the tRNA in the A site. The ribosome catalyzes bond formation. Now the chain has one more amino acid. Thirdly, translocation occurs. The P site tRNA now leaves the ribosome, and the ribosome moves the remaining tRNA carrying the growing polypeptide to the P site. The mRNA and tRNA move as a unit. This movement brings into the A site the next mRNA codon to be translated, and the process can start again with step 1. So as your slide points out, there are 3 compartments for elongation: the A site, the P site, and the E site. The A site binds the incoming charged aminoacyl tRNAs. The P site binds the tRNAs carrying amino acids that have formed polypeptide bonds with the growing polypeptide chain but have not yet dissociated from their corresponding tRNA. And the E site releases the dissociated tRNAs so that they can be recharged with free amino acids. Moving on, elongation continues until a stop codon reaches the ribosome's A site. The stop codons UAA, UAG, and UGA do not code for amino acids, but instead tell the translation to stop. The completed polypeptide, typically several hundred amino acids long, is freed, and the ribosome splits into its respective subunits. And the cartoon on your slide details this process. And this next slide summarizes the central dogma of molecular biology. Genetic information in DNA is transcribed into RNA and then translated into polypeptides. But how do these processes occur? Transcription and translation are linguistic terms, and it is useful to think of nucleic acids and polypeptides as having languages. To understand how genetic information passes from genotype to phenotype, we need to see how the chemical language of DNA is translated into the different chemical language of polypeptides. What exactly is the language of nucleic acids? Both DNA and RNA are polymers made of monomers strung together in a specific sequence that convey information, much as specific sequences of letters convey information in English. In DNA, the monomers are of 4 types of nucleotides, which differ in their nitrogenous bases, A, T, C, and G. The same is true for RNA, although it uses the base U instead of T. The language of DNA is written as a linear sequence of nucleotide bases, and you can see the blue strands on the left-hand side of the diagram labeled transcription represent DNA. Specific sequences of bases, each with a beginning and an end, make up the genes on a DNA strand. A typical gene consists of thousands of nucleotides, and a single DNA molecule may contain thousands of genes. When a segment of DNA is transcribed, the result is an RNA molecule. This process is called transcription because the nucleic acid language of DNA has simply been rewritten or transcribed as a sequence of bases of RNA, the language that is that of the nucleic acids. The nucleotide bases of RNA molecules are complementary to those on the DNA strand. And on the transcription part of this cartoon, we see the growing RNA molecule represented as a red line. And on the center segment of our cartoon, we see RNA processing. We were talking about that a few slides ago, where we see the primary RNA transcript on the top. You've got an exon, an intron, an exon, an intron, and an exon. And we see that the introns are spliced out, and the molecule is capped, and it also is tailed. Translation is the conversion of the nucleic acid language to the polypeptide language. Like nucleic acids, polypeptides are polymers, but the monomers that make them up, the letters of the polypeptide alphabet, are the 20 amino acids common to all organisms. Again, the language is written in a linear sequence. In this case, the sequence of nucleotides of the RNA molecule dictates the sequence of amino acids of the polypeptide. But remember, RNA is only a messenger. The genetic information that dictates the amino acid sequence originates in the DNA. What are the rules for translating the RNA message into a polypeptide? In other words, what is the correspondence between the nucleotides of an RNA molecule and the amino acids of a polypeptide? Well, keep in mind that there are only 4 different nucleotides in DNA: A, G, C, and T. And there are only 4 different kinds of nucleotides in RNA: A, G, C, and U. In translation, these 4 must somehow specify 20 amino acids. If each nucleotide base coded for 1 amino acid, only 4 of the 20 amino acids could be accounted for. In fact, triplets of bases are the smallest quote-unquote words of uniform length that can specify all the amino acids. There can be 64, that is 4 to the third, possible code words of this type. That is more than enough to specify the 20 amino acids. Indeed, there are enough triplets to allow more than 1 coding for each amino acid. For example, the base triplets AAA and AAG both code for the same amino acid. Experiments have verified that the flow of information from gene to protein is based on a triplet code. The genetic instructions for the amino acid sequence of a polypeptide chain are written in DNA and RNA as a series of 3 base words called codons. 3 base codons in the DNA are transcribed into complementary 3 base codons in the RNA, and then the RNA codons are translated into amino acids that form a polypeptide. So as this cartoon summarizes, 1 DNA codon, that is 3 nucleotides, moves into 1 RNA codon, that is 3 nucleotides, and that moves into 1 amino acid. So here we'll quickly summarize the central dogma of life, or the central dogma of molecular biology. It starts with the DNA molecule. Transcriptional machinery comes in, and it creates what we call an mRNA. The RNA is processed and packaged so it can move out into the cytoplasm. Once it moves out into the cytoplasm, it joins with the ribosome and is translated into a polypeptide or a protein. Moving on, since discovering how genes are translated into proteins, scientists have been able to describe many heritable differences in molecular terms. For instance, sickle cell disease can be traced to a change in a single amino acid in one of the polypeptides in the hemoglobin protein. This difference is caused by a single nucleotide in the coding strand of DNA. Any change in the nucleotide sequence of DNA is called a mutation. Mutations can involve large regions of a chromosome or just a single nucleotide pair, as in the sickle cell allele. Occasionally, a base substitution leads to an improved protein or one with new capabilities that enhance the success of the mutant organism and its descendants. Much more often, though, mutations are harmful. Let's consider how mutations involving only 1 or a few nucleotide pairs can affect gene translation. This next slide is simply to describe to you how a point mutation, that is, a mutation of 1 single base pair, can drastically affect the central dogma. So let's think about this critically. The sequences that we see on the screen are DNA sequences. How can you tell? Well, remember, the DNA sequence contains the base pair T. If this was RNA, we would see the base pair U in place of the T. So let's say, for argument's sake, that the sequence that we see on the screen is the template strand. So the transcriptional machinery will come in and start transcribing this DNA sequence into mRNA. So the first set of triplets, TAA, would transcribe into AUU. The second set of triplets, CTG, would transcribe into GAC. GAC, when translated into an amino acid, would be aspartic acid. Now, if we look at the bottom half of the cartoon, we see what we call a point mutation. That is, only 1 base has changed. So we still start with the triplet TAA, which is going to be transcribed into AUU, But now, instead of CTG, we have CCG. And CCG will be transcribed into GGC, which is glycine. So you can see how that one small mutation on only one base changes the amino acid. So you go from the original sequence, aspartic acid, to the mutated sequence, which codes for glycine. Moving on, mutations within a gene can occur as a result of base substitutions, deletions, or insertions. A base substitution is the replacement of one base or nucleotide by another. Depending on how a base substitution is translated, it can result in no change in the protein, in an insignificant change, or in a change that might be crucial to the life of the organism. Because of the redundancy of the genetic code, some substitution mutations have no effect. For example, if a mutation causes an mRNA codon to change from GAA to GAG, no change in the protein product would result because GAA and GAG both code for the same amino acid. Such a change is called a silent mutation. Other changes of a single nucleotide do change the amino acid coding. Such mutations are called missense mutations. For example, if a mutation causes an mRNA codon to change from GGC to AGC, the resulting protein will have serine instead of glycine at the position. Some missense mutations have little or no effect on the shape or function of the resulting proteins, but other, like in the case of sickle cell disease, can cause changes in the protein that prevent it from functioning normally. Some base substitutions, called nonsense mutations, change an amino acid codon into a stop codon. For example, if an AGA or arginine codon is mutated to a UAG or a stop codon, the result will be a prematurely terminated protein which probably will not function properly. Mutations involving the deletion or insertion of one or more nucleotides in a gene often have disastrous effects. Because mRNA is read as a series of nucleotide triplets during translation, adding or subtracting nucleotides may alter the triplet grouping of the genetic message. All the nucleotides that are downstream of the insertion or deletion will be regrouped into different codons. For example, consider an mRNA molecule containing the sequence AAG, UUU, GGC, GCA. This codes for lysine, phenylalanine, glycine, and alanine. If a U is missing in the second codon, as a result of a deletion in the DNA, the resulting sequence will be AAG, UUG, GCG, CA, which codes for lysine, leucine, and alanine. The altered polypeptide is likely to be non-functional. Inserting one or two nucleotides would have a similarly profound effect. I'd also like to throw out the vocab word mutagen here. So what causes mutation? Mutagenesis, the creation of mutations, can occur in a number of ways. Mutations resulting from errors during DNA replication or recombination are known as spontaneous mutations, as are other mutations of unknown cause. Other sources of mutation are physical and chemical agents called mutagens. The most common physical mutagen is high-energy radiation, such as X-rays and ultraviolet light. Chemical mutations are of various types. One type, for example, consists of chemicals that are similar to normal DNA bases, but that base pair incorrectly when incorporated into DNA. Many mutagens can act as carcinogens, agents that cause cancer. What can you do to avoid exposure to mutagens? Several lifestyle practices can help, including wearing protecting clothing and sunscreen to minimize direct exposure to the sun's UV rays and not smoking. But such precautions are not foolproof, and it is not possible to avoid mutagens such as UV radiation and secondhand smoke entirely. Although mutations are often harmful, they can also be extremely useful both in nature and in the laboratory. Mutations are one source of the rich diversity of genes in the living world, a diversity that makes evolution by natural selection possible. Mutations are also essential tools for geneticists. Whether naturally occurring or created in the laboratory, mutations are responsible for the different alleles needed for genetic research. So let's do a quick recap of tonight's lecture. You should be able to outline the basic steps of DNA transcription into RNA. You should know what the key difference between pre-RNA and mRNA is. You should be able to list the components of translation. You should know what the genetic code is, and you should know its key features. You should be able to outline the basics of translation of RNA into proteins. You should know the central dogma of life, or the central dogma of molecular biology. You should know what a mutation is. You should know how mutations occur, and you should be able to list the major types of DNA mutations.